I'd like to start off by thanking Merck & Company for endowing the Scolnick Prize in Neuroscience. It was endowed by Merck in honor of Ed Scolnick, who served for 17 years as head of Merck Research Labs. Now, as many of you know, the endowment has grown in value over the years and now consists of this fantastic McGovern Institute mug, which will be presented to the prize winner tonight. And happy April Fools. I am very, very happy to say that this year's prize winner and the future mug owner is Tom Jessell, the Claire Tow Professor of Biochemistry, Molecular Biophysics, and Neuroscience at Columbia, the co-director of the Kavli Mind-Brain Initiative at Columbia, and Howard Hughes investigator. Tom received his PhD from Cambridge University and has spent most of his professional career at Columbia. He's a member of the U.S. National Academy of Sciences, member of the Royal Society, and he's won many other awards for his work, including the Bristol-Myers Squibb Award, the Kavli Prize, and the Gairdner Award. Now, there's probably no more fascinating or important problem area in neuroscience than how the brain forms during development. How does an embryonic cell know what kind of neuron to become? How does it integrate itself into local neural networks? And how does it form all the appropriate connections with neurons throughout the brain so that it can mediate our great behavioral complexity? Tom Jessell has used the spinal cord in this work as a model system. He's used it because of its simplicity and the evolutionary conservation, which makes it really an ideal system for studying the general principles of neural development. As just a few examples, he discovered the morphogens, the diffusible molecules that determine whether cells in the spinal cord become sensory-related neurons in the dorsal cord or motor-related neurons in the ventral cord. He's discovered the master regulators in the Hox gene family that determine how cells acquire their particular properties and all their central and peripheral connections. And he's used this knowledge to manipulate the different cell groups to understand that link between the functional neural circuitry and motor behavior. He's also used his knowledge of the transcriptional regulation of cell fate to create different neuronal subtypes from embryonic stem cells, transplanted them into the spinal cord where they do form the appropriate neural connections. Now, the work has obvious, tremendous implications in clinical neuroscience, most particularly in potentially treating motor neuron disease in the future, but I think all of us can imagine tremendous, widespread clinical applications of this sort of work in the future. We are delighted to have him here as our prize winner today. We'll talk about shifting circuits for motor control.
Well, thank you, Bob, for that introduction. It really is a threefold privilege to be here this afternoon. The first reason is just because it's MIT. And in the context of neuroscience, MIT is one of those places that you pay attention to. Actually, about a year ago, Nancy was kind enough to host a bunch of people, including architects who are interested in developing Columbia's own little neuroscience venture, and Nancy showed us around the epicenter of Bostonian neuroscience. I can say that with Clifford looking sternly on here. So just to be here is a real thrill. The second reason, because this is a Scolnick lecture, and I regard myself in a funny way as a sort of great nephew once removed of Ed, in the sense that my doctoral thesis advisor was Leslie Iverson, who went to work for Merck at Turlings Park. And as Les writes, one of his pet projects was the development of drugs based on substance P pharmacology, and he points out that Ed was a real champion in that. and I did my thesis on substance P and its role in sensory transmission. So it's a privilege to be here on that context. And finally, I regard MIT as one of the meccas of motor control, with Emilio and others before him. And so Emilio has been arguing for many years about the primacy of limb and muscle biomechanics and synergy groups, and us dumb molecular biologists have taken a while to appreciate the importance of this in the context of thinking about the organization of motor systems. So a little bit of what I want to do this afternoon is to try and convince Emilio, and perhaps you, that we've finally seen the light, and that we, from this reductionist molecular perspective, are beginning to appreciate those aspects. So it's an exciting time for the motor system, I think, as I'll try and convey. And the last thing, in the context of Bob's introductory remarks, if you leave this lecture theatre with no other change in your thought, it's to try and convince you that the spinal cord is not actually a simple system. It's damn complicated, and we don't understand it, and maybe we'll never understand it, but it's what drew us into the system in the first place, so we're stuck with it. So what I want to talk about this afternoon is really to try and approach the systems and circuits that give animals the ability to act on demand. That is, to process the sensory world that surrounds them, to implement their inner intent, if you like, to purposeful action through movement. And the simplest of movements engage circuits at many different levels of the central nervous system. The planning of actions and movements prior to overt signs of this are thought to be in part the work of circuits in cortical centers and in other supraspinal centers. But from the time of Sherrington onwards, about 100 years ago, we've come to realize that the executive component of movement, deciding just which muscles to activate at which time and in which order and which sequence, though the problems had in large part have been assigned to the circuits that are resident in the spinal cord. So at one level, if one wants to try and study aspects of the expression of behavior, that problem reduces to knowing how the nervous system engages muscles in this precise pattern and in a context and task-dependent manner. And within the spinal cord, some of the most sophisticated forms of motor behavior are achieved through the mechanics of the limb, whether it be the iterative activation of flexor and extensor muscles during locomotion, or the more complex arm and hand synergies and manipulations that are involved in reaching and grasping and retrieval. These are some of the features of limb mechanics devoted to the motor system. And I think it's fair to say that neither in locomotion nor in reaching behaviors do we really have a very firm grasp of the circuits that underlie these behaviors. And so this is what I want to try and introduce to you this afternoon from this molecular perspective. So just to introduce all of you, should I?
Before you start,
Yeah,
just
all right.
that one.
Okay.
This one's going to feed back, and if you just want to speak for a second, we can just hear it in the room because you're feeding back a little.
Okay, is that better?
That's probably
Great, thank you. So this, in very simplified schematic form, is a cross-section through the spinal cord that introduces some of the neuronal elements that are controlling movement. Motor neurons generated in the ventral spinal cord innervate individual limb muscles, and a variety of other inputs. So the problem here is really reduced to twofold. How do motor neurons select muscle targets in a way that conforms to the biomechanical demands of the limb? And the second problem is, how do the many inputs to motor neurons organize themselves in a way that makes sense in terms of controlling motor neuron firing And what is shown here are, in schematic form, sets of interneurons that serve as local controls of motor output, sensory feedback, monitoring the state of muscle contraction and feeding that in its simplest form in a monosynaptic connection with the motor neurons. And not shown here are the descending systems that engage these various segmental spinal circuits. And the output here, as shown in this EMG recording, is that a given flexor and extensor muscle, for example, at a given joint, are activated in this precise alternating phase. So lest you think that this binary activation is all that the spinal cord can do, if we consider, for example, the locomotor step cycle, then the limb, a typical mammalian limb, contains some 50 to 60 different muscle groups. And the activation of these muscles during the lift and swing and stance phases of one step here involves the synchronous recruitment, and this is from work by Trevor Drew, of many different muscles in this precise temporal order. And that these muscles can be classed at individual joints into antagonists, as well as into muscles that serve a common function in controlling the flexion or extension of an individual joint, the joint synergists here. So somehow these circuits in the spinal cord and descending systems have to engage this complexity of muscle organization. So in this afternoon's talk, I really want to take these basic features of spinal motor circuit organization to ask 2 and perhaps 3 questions. And importantly, work by Eric Frank and others have indicated that the final pattern of connections in the spinal cord, at least from the sensory motor control, is achieved in an activity-independent manner. And that begins to suggest that there may be molecular determination of the circuits that we're looking at in these diagrams here. So this afternoon, I want to address 2 questions. If we knew what the molecular grain of neuronal subtype differences were in the spinal cord, could we use that to infer strategies for specificity in the wiring of motor circuits? A developmental question. Could we also use cell type specific patterns of gene expression to interrogate motor behaviors one neuronal class at a time, providing access that would be difficult to achieve in other ways? And lurking underneath these 2 questions is this issue of what really are the limits of approach using these molecular techniques if one wants to approach motor systems at this systems level. And on the way, perhaps I'll indicate where molecular approaches have got to, at least today. So the first question one can ask is, what is the molecular grain of information that is relevant to the specification and organization of neurons in the spinal cord? And work that we've done over the last 2 decades or more have told us that there's one general class of proteins that are particularly informative in revealing neuronal subtypes, and they turn out to be transcription factors, DNA binding proteins that are expressed in a highly cell-specific manner. And so we're looking here in red at motor neuron progenitors and in green at postmitotic motor neurons expressed and generated in one domain of the spinal cord. And work that I'm really just going to summarize for its sort of general principle here is that motor neurons are generated in the ventral spinal cord together with 4 major classes of interneurons that conspire to control motor output. And the different identities of these neurons, both in progenitor stage and in postmitotic neurons, can be recognized by individual sets of transcription factors. And these transcription factors are patterned here in response to an extrinsic inducing signal, in this case the morphogen sonic hedgehog. So if transcription factors control neuronal identity, how do you make the link between the identity of a neuron and the connection of those neurons through synaptic contacts with partners to wire circuits? And this raises the general question of the link between identity and connectivity, which I want to address for the first part of this talk. And so it's clear that identity controls connectivity. We've shown that. Other people have shown that. How is it achieved? So one prominent view is a sort of Sperrian view that identity gives neurons different surface labels, and those surface labels can be recognized by incoming axons to distinguish one cell type from another. And while this seems a compelling and plausible idea. In fact, in the mammalian nervous system and even in flies and worms, it's been remarkably difficult to identify the nature of these synaptic recognition determinants. We've known for an even longer time, from the time of Ramon y Cajal, that neurons with a common projection pattern and common input are not scattered randomly throughout the central nervous system, but are clustered together in highly stereotypic positions from animal to animal within a species and across related species. So this emphasizes the role of neuronal position as an important consequence of neuronal identity in parallel with these potential surface labels. And in our mind, it raised the issue of what is the relative contributions of label versus location in the establishment of connections. And one reason, from a narrow focus in the spinal cord, of paying attention to neuronal position comes from looking at the organization of motor neurons in relation to the muscles in the limb that they innervate. And this was a set of observations that was first made by George Romanes, now 70 years ago, looking at the organization of motor neurons and muscle. And so this is a typical mammalian limb. This would be proximal, distal hind limb, so this would be the foot, this would be the ankle joint, the knee joint, the hip joint there. And we've known for many years that motor neurons that innervate an individual muscle in the limb are not scattered but are clustered into so-called motor neuron pools. And this just shows that this transcriptional specification of neuronal identity extends to each of the 50 motor neuron pools that are found in a typical mammalian hind limb, in this case the mouse. But what Romanes pointed out is that the motor neurons clustered into a pool, the motor neurons that are innervating related synergist muscles at a particular joint, are then tightly clustered with their neighbors innervating, in this case, ankle flexors, to produce a higher order organization, which we've termed a columel, or a little column here. And if you look more generally at the organization of these columels within the spinal cord, you can see this precise relationship between the dorsiventral and mediolateral position of these motor neuron clusters and the limbs that they innervate. So the most dorsally located motor neurons are going to innervate the foot, the next most ventral, the ankle, the knee, and the hip. And this is a remarkable feature of organization that is conserved from frog to man in vertebrates. So it must mean something here. So what does this mean in terms of the organization of connections? So one class of cells that clearly respect this columellar synergy organization are the sensory neurons that provide feedback information onto motor neurons. So from the classical studies of Eccles, where they've used physiology to map the specificity of connections between a class of proprioceptive afferents and their cognate motor neurons, what Eccles and his colleagues showed is that monosynaptic connections are formed in an extremely strong, robust manner with homonymous motor neurons, that is, motor neurons projecting to the same muscle, but there are also significant excitatory input into synergist motor neurons innervating synergist muscles, but that these one set of afferents take pains to avoid forming contacts with antagonist or unrelated motor neurons. And when you plot this out as a matrix, you can see evidence of this synergy group there, stimulating one sensory afferent forms connections at different weighted strengths with 4 different motor neuron pools. So the problem from a developmental perspective is how one class of sensory afferents chooses among 50 to 60 different motor neuron sets, 3 or 4 to form connections with precisely invariant weights, which are necessary to control aspects of limb movement. And so we were interested in testing the possibility here that the motor neuron position, which is so tightly related to limb muscle and sensory input, might have something to do with this specificity process. And so in order to do that, we wanted to do, in this case, initially, a sort of thought experiment. This is the normal organization of sensory afferents innervating different pools and columels along the dorsiventral axis. What happens if we could strip motor neurons of their molecular identity, but still get them to innervate individual muscle targets, and then look at the specificity of innervation by proprioceptive afferents? If there was mayhem ensuing once we stripped them, then this would argue that perhaps the surface label expressed was important. But if we saw an underlying order here, then perhaps it would suggest that the position of these neurons, rather than their surface label, might be an important determinant. And because we know the transcriptional specification of these motor neurons at limb levels of the spinal cord, 21 of the mammalian Hox genes are devoted to that, it gives us a way genetically of trying to inactivate motor neuron identities. We can't knock out 21 Hox genes, but what we can do is knock out an essential Hox cofactor that is required for the output of all 21 Hox genes. And when we do that, I'm not showing you, what we find is that we completely ablate the subtype identity of motor neurons, yet they innervate muscle targets, albeit at random. And you can see that the cell bodies of motor neurons that innervate a given muscle target are no longer tightly clustered, but are scattered at random amongst the general cohort. So this gave us the ability to look at the sensory connectivity to motor neurons as a function of their position in the spinal cord. And because this work is published, I'm just going to summarize the conclusions of this, which are shown most clearly here. So we have this matrix of motor neuron positions. If we strip motor neurons of their identity, or if we in fact get rid of motor neurons completely, what happens is the proprioceptive afferents will grow to their normal dorsoventral mediolateral position, completely independent of the subtype identity or presence of motor neurons themselves. So there is a sensory tier targeting system, which is independent of motor neurons, which gets sensory afferents into the right position. And when viewed from that perspective, it begins to make sense of why evolution has taken such pains to position motor neurons in a way that conforms to their muscle targets. Because unless you put motor neurons in the right position, they don't have a hope of being innervated by the sensory afferents that are going to grow to that coordinate position. So this began to produce the idea that motor neuron position is important because it puts neurons in a position to receive appropriate sensory input. Turns out that this is not a particularly new idea, although it surprised us, because the initial innervation of skeletal muscles in the limb by motor neurons operates along much the same principle. The muscle target doesn't provide information. It's the limb mesenchyme that positions axons and controls muscle cleavage in a way that by the time the axons get to a coordinate position, there's only one muscle that's cleaved and waiting for them there. And in fact, in flies, from work by Mike Bate and by Wes Gruber, a very similar sensory targeting scheme. So it begins to raise the possibility that these target-independent guidance cues impose the requirement for neuronal clustering and neuronal positioning to achieve aspects of connectivity. And in our mind, this raised the question of whether this form of positional information is true not just for sensory motor connections, but for the connections of local microcircuits in the spinal cord formed by interneurons. So I'm going to spend a couple of minutes providing evidence that this is a general principle of microcircuit assembly within the mammalian spinal cord. And so I pointed out earlier that in addition to motor neurons, there are 4 major classes of interneurons. There are 2 features here that are important. One is that we know that all motor neurons derive from a single domain, but can diversify into 50 or 60 different subtypes. So is the same true for interneurons, that these major classes can be fractionated into many different subtypes that perhaps might get us closer to the grain of organization of the motor system. And so this is just the major cardinal subtypes distributing themselves. What happens if we focus on one of these subtypes and ask how many different fractionated subgroups can we identify? And this is work that Jay Bykoff, a postdoctoral fellow in the lab, has been performing over the last couple of years. And he focused on one set of these interneurons, the so-called V1 interneurons, which turn out to be inhibitory interneurons and correspond to many of the better characterized interneuron subtypes, the 1A reciprocal inhibitory interneurons and the Renshaw cells in the spinal cord. And so Jay, because he knew that the engrailed 1 gene defined this subtype, was able to make an engrailed 1 Cree-Rosa cross, label all the V1 neurons, independently used a different transcription factor to label a set of GABAergic inhibitory interneurons in the dorsal spinal cord, and did a molecular comparison to find genes that are expressed in the V1 but not in these DI4 interneurons. And from that, Jay's work identified 18 transcription factors that divide the V1 cohort into something greater than 25 different subclasses of interneurons. Since there are 4 classes of interneurons, if the same degree of diversity operates, it poses the rather daunting issue that there are at least 100 different molecularly defined subtypes of interneurons sitting in the spinal cord ventrally, presumably to control motor output. And these are just a couple of examples. So in green are all of the V1 neurons labeled in this genetic trick, and in red are the co-labeled cells defined by these transcription factors. And what you can see is that some of this co-labeling occurs in the dorsal region and some of it occurs in the ventral region, a point I'll come back to. So the first question you might ask is, why do you bother to make this vast diversity of interneuron subtypes? And we don't know, but a couple of speculative thoughts. One might be that there are a lot of motor neurons, so maybe you generate an equivalent diversity of interneuron subtypes to accommodate each of the different motor neurons. Second way of thinking about this comes from this sort of diagram, that these interneurons serve essential functions in mediating descending control of motor output. So you could imagine where 4 different varieties of these V1 interneurons innovate essentially the same set of motor neurons in terms of their limb innovation, but each one of these is designed to receive input from a different class of descending fiber to give these descending systems a labeled independent line control of motor output. So by the time you do this and then form this to different classes of interneurons, it begins to be plausible as to why you'd need this diversity to accommodate the different descending controls and sensory control of motor output. We don't know that, but we're sort of interested in testing this idea. But the major point I want to emphasize in this section is that when you start to look at these 18 different subsets in some detail, you find something that looks rather like the motor neuron system, where motor pools are arrayed along the dorsiventral and mediolateral axis. We find individual subsets that are arrayed along the mediolateral axis, and as shown here for 3 of these transcriptionally defined V1 subsets, the epicenter concentration of these neurons is neatly arrayed along the dorsiventral axis. So now we have interneuron subtypes located along the dorsiventral axis, motor neuron subtypes located and arrayed along. Is there any relationship? And does the spatial relationship between motor neuron and interneuron constrain the organization of the microcircuit? And remember that since dorsiventral axis in the spinal cord corresponds to proximal distal axis in the limb, we're talking about a system that might be dedicated for different circuits for the control of muscles at different joints. So just to illustrate that this is in fact the case, I want to focus on a set of interneurons that are V1 derived that express this MAF-A transcription factor that are located in the extreme ventral region of the spinal cord. So it turns out that the MAF-A neurons correspond to this classical recurrent inhibitory interneuron, the Renshaw cell here. So the basic Renshaw cell circuit has a motor neuron projecting to muscles, sending a collateral that recruits this inhibitory interneuron, which feeds back onto the motor neuron, presumably to truncate the period of muscle activity. There's been confusion in the literature as to whether Renshaw cells, like motor neurons, also receive sensory input or not. Some say no, some say yes. So could that ambiguity or uncertainty reflect precisely which sensory neurons are projecting along the dorsiventral axis, and perhaps only some of them are capable of recruiting and innervating Renshaw cells. And so Jay has done a series of anatomical studies that really confirm that view. So remember that the gluteus motor neurons settle in a ventral region, therefore their sensory afferents go ventral, tibialis more dorsally, foot even more dorsally. And what we find when we label the Renshaw cell dendrites with a motor neuron marker, when we label the Renshaw dendrites with calbindin as well as with MAF-A and look at sensory input there, we're using a vesicular glutamate transporter which labels the sensory terminals, and defining the muscle of origin by labeling the muscle with CTB so it's transported into the central terminals. The gluteus afferents co-express VGLUT1 and CTB in this sort of cyan color and innervate motor neurons. Innervate Renshaw cells. Half the Renshaw cells are innervated by gluteus afferents, many fewer by tibialis, and none by intrinsic foot sensory afferents. So what you find here is a position-dependent incidence of innervation of Renshaw cells by sensory afferents there, arguing that for this class of interneurons, only certain sensory afferents that are normally going to project in the ventral region have a hope of innervating. And this is true not just for this aspect of Renshaw cell circuitry, but for the recurrent loop itself. And so this is supported by physiology, where when we record together with George Mentis's group at Columbia from Renshaw cells, we find that gluteus forms strong excitatory inputs, very few inputs from tibialis, and no monosynaptic inputs from the intrinsic foot. So what seems to be happening in this situation is you're building microcircuits as a function of neuronal settling position in the spinal cord that are dedicated according to the muscles that are going to innervate at different joints. So the hip joint innervation here, the motor neurons that innervate the hip, form this classical recurrent inhibitory circuit as well as having sensory input, and at the opposite extreme, the foot motor neurons never bother to give off a collateral. Renshaw cells never bother to inhibit motor neurons, and there's no sensory input. So these and other studies on different classes of these V1 are beginning to argue that there's no such thing as a canonical motor neuron circuit in the spinal cord, but that circuit depends on the muscle mechanics that they're going to innervate, and by inference along the dorsoventral axis of the spinal cord. So why would you bother to design these different circuits? Well, one possibility is, from limb mechanics, the degrees of freedom at different joints vary rapidly. So if you look at the elbow, you're basically operating in one plane, whereas the hand has many degrees of freedom. So maybe in order to meet those functional demands of muscles at different joints, you have to build different circuit organization. So if we come back to the position versus label issue, at least from these studies in the spinal cord, it seems likely that neuronal position has as important a role in determining the final pattern of connectivity as any of the presumed surface labels, most of which are still remain to be identified. So where this shakes down in terms of the relative contribution of label and location, we're in the middle of finding out. And in the spinal cord, position in progenitor cells along these gradient morphogen axes gives you identity. Identity then translates back into a new position of motor neurons, which then determines connectivity. So position and connectivity patterns are intricately linked in the spinal cord. And if this is true in the spinal cord, then it seems at least plausible that it may be true in other regions of the CNS. So when you look, for example, at these classical homuncular maps, not that they actually represent anatomical perfection, it's hard not to believe that the relationship between the organization of motor cortical neurons or somatosensory cortical neurons and the periphery there might not have similar functions. Or if you look at the different subnuclei of the amygdaloid complex, which look just like motor pools, why are you clustering these neurons in a way if you're not using it to simplify the problem of connectivity? So we're interested in exploring these ideas in addition in the spinal cord, but also seeing how well they extend to other regions of the central nervous system. So this is one developmental example of how you can use neuronal identity to tease out principles of circuit construction, in some cases surprising principles, such as the emphasis on neuronal location. For the rest of the talk, I want to move to the idea that neuronal identity can also be useful in extracting features of the relationship between motor circuit organization and limbed motor behaviors. And much of the work in rodents has traditionally been focused on the locomotor cycle, the step cycle here, whereas the work in primates and humans has tended to emphasize this evolutionary, more sophisticated aspect of object manipulation and retrieval and grasp. And so these are highly specialized processes giving rise to this sort of extraction of this quote from Byron, the slight chance of reaching. These skilled tasks have an inherent failure component in there. So can you even begin to approach problems of reaching in rodents like mice? And so Ian Wishaw's group has made an argument that there are conserved modules of reaching and that there's a sort of a breakdown of these different components that can be analyzed from rodent to primate. So the initial lift of the hand and the limb, the advance, the pronation, and then the grasp of a target retrieval here. So we've been interested in asking whether, in fact, we can use the mouse to study aspects of skilled reaching and then begin to probe elements of the circuit organization. And in one view, we're trying to deal with the problem that motor systems have been advertised, the rapidity and refinement during reaching tasks. So, as you know, the idea of planning motor commands and activating muscle, eventually producing a sensory feedback which is relayed back into the central nervous system, arrives too late to do much good in terms of the online updating of a motor plan. So the nervous system has designed shortcuts, if you like, to deal with this so-called delay problem. One is to have a copy of that motor command transmitted at various regions in the central nervous system to provide more rapid update. And when that copy is integrated with the actual sensory feedback, these systems serve as comparators to modify and monitor errors between planned and actual motor performance there. And a second system that has been used is to use these local segmental sensory gates further down in this system to modify the information transfer between sensory and motor. And so what I want to do is give you examples of both the way in which an analysis of circuitry might provide insight into the copy and gate system during a skill reaching task in the mouse. And so these are studies performed initially by Ayman Azim, who set upon the challenge of trying to develop these motor reaching tasks in mice. So what he did is he took a mouse, separated it from a food pellet by a thin perspex slit, and then monitored the kinematics of limb trajectory as it reached for this target with a series of cameras at different angles. And so we could visualize the forepaw with a marker and the food pellet target and monitor this in kinematic time. And I'll just give you one example of one of these reach tasks. So here is a mouse reaching for a pellet through the slit, and we can monitor a variety of parameters, the trajectory, the velocity of the forepaw as it's reaching for this task in a way that is at least beginning to resemble the sorts of analyses and studies that have been done in primate systems there. And the first conclusion was that independent in wild-type animals of the success of actually grabbing the food pellet, the trajectories and velocities of the forelimb during these tasks, whether it's a hit or a miss, were remarkably similar. And this is just shown here. So in the end, the successful reach is largely a part of the late-stage pronation and much less a part of the initial reach trajectory. So Ayman convinced himself that this sort of behavioral assay might be amenable to the dissection of certain elements of motor circuits at the forelimb level. And then the question is, which circuits do you study? Where do you begin to tackle this very complicated program of motor output? And in this consideration, we were intrigued by studies that have been carried out for the last couple of decades by Bror Alstermark in Sweden and Tadashi Isha in Japan, who focused on a set of propriospinal neurons that are found at cervical levels of the spinal cord that have a last-order premotor excitatory input to forelimb level motor neurons that are controlling limb muscles here. So this, in principle, might provide one way of looking at the last-order input here. And Alstermark and Isha's studies have shown that when you sever this descending branch, you get defects in the reach, but not in the grasp or retrieval, supporting the idea of the modular organization of reach in mammals. What intrigued us more was this second branch of the same neuron that is ascending the spinal cord, ending in the lateral reticular nucleus, a prenucleus, which then provides output to the cerebellum. So this branch here then might begin to look like a copy of a motor command being delivered to motor neurons here. And this is a somewhat strange organization compared, for example, to the traditional motor efference copy systems that have been identified in the spinal cord. For example, the ventrospinal cerebellar tract here, which receives a copy, so the story goes, of input to motor neurons and relays that up to the cerebellum. Here we have a variant of this where there's a similar projection to the cerebellum, but it's welded together with input to motor neurons. And so in this traditional system, it places an emphasis on the information being provided to the VSCT neurons being exactly the same as the information to the motor neuron to get an accurate output or an accurate internal copy, if you like. Whereas in this system, even though this is not the only input to motor neurons, you're pretty sure that the information being transmitted to the cerebellum is a copy of the information being transmitted. So maybe in order to achieve the sophistication of motor control, you have to design these internal copies through a variety of different mechanisms. So we were interested on the basis of Ulstermark's work, and we've been collaborating with Broer in these studies, in seeing whether actually equivalent propriospinal neurons even exist in the mouse. And we have some evidence from physiology that they exist, because if we record from a propriospinal neuron and antidromically activate by stimulation of the lateral reticular nucleus, as well as from motor neurons, you can get a classical antidromic spike collision, arguing that there are neurons in mouse cervical spinal cord that project in both directions to both target regions. We were more interested in the idea, can we identify molecularly these propriospinal neurons and use that to begin to manipulate this set of neurons, in particular to try and ask whether this ascending internal copy has any role in calibrating or modifying motor control. And so the feature of these neurons from this molecular perspective is that these are ipsilateral, last-order, excitatory premotor interneurons here. And of these different interneuron subtypes, the only class that conforms to that anatomy are these so-called V2A neurons, which can be defined by a transcription factor CHEX10. So we then ask the question, amongst the CHEX10 V2A neurons, do any of them conform to the propriospinal neurons defined by a projection to the lateral reticular nucleus and to motor neurons? We know that V2A form these inputs to motor neurons. Do they also project to the lateral reticular nucleus? So to do that, we made a mouse in which CHEX10 drives Cre recombinase, and then we injected an adeno-associated virus encoding both channel rhodopsin and a fluorescent protein into cervical levels of the spinal cord. Here the fluorescent protein is important. If we look in the lateral reticular nucleus, you can see a very dense innervation of the lateral reticular nucleus neurons by neurons that are V2A derived that originate from cervical spinal cord. So this says that as a population, there are neurons that project to the lateral reticular nucleus and to motor neurons, but does an individual neuron have this bifurcating innervation pattern? And to test this, we again labeled these neurons using the CHEX10 Cre control. We injected CTB to retrogradely label these neurons from the lateral reticular nucleus. We injected wheat germ agglutinin into forelimb muscles. It's transported into motor neurons, hops into the V2A neuron, and then we asked whether there were neurons that are green, wheat germ positive, and CTB positive. So here's the green neuron, it's CTB positive, is wheat germ positive, you overlay. And what emerged is about 1/3 of all of the V2A neurons conform to propriospinal neurons by these anatomical criteria. So if we can identify these neurons, can we now manipulate their function and ask whether there's any impact on reaching behavior? And so we've done this in two ways. I'll emphasize one of these ways. So by virtue of the fact that we can identify V2A neurons, we can express a diphtheria toxin receptor in those neurons. We can then inject diphtheria toxin and kill those neurons very effectively. And we see dramatic errors in reach trajectory. The problem is that only 1/3 of the V2As are propriospinal, and by killing the neurons, we block the premotor output as well as this more interesting ascending internal branch here. So we wanted to do something that allowed us to focus on this branch of these neurons with a greater degree of selectivity, and in fact focus only on that 1/3 of the total set of V2A neurons. And so what we've been trying to do is find ways of selectively initially activating but eventually inactivating this ascending branch and look at the consequences for motor control to address this sort of internal copy route here. So the way we've tried to do that is again in the CHEX10 Cre mouse to inject channel rhodopsin into these V2A marked neurons and then place a light probe and photoactivate the terminals of these V2A neurons in the lateral reticular nucleus. Now, the first thing one would need to worry about here is that there are many instances where terminal activation with optogenetic methods results in back-propagating spikes, and then we might activate the motor system. So Eyman and Broer tested this very rigorously, and while local electrical stimulation in the lateral reticular nucleus is sufficient to activate antidromically the propriospinal neuron, no amount of photoactivation over a wide range of stimulus parameters ever resulted in any activation of the propriospinal neuron. So we conclude, at least at this level, that in this, as in many other neurons, photoactivation of terminals doesn't result in back-propagating spikes. Yet we know that photoactivation is very effective in triggering the activity of the target lateral reticular neurons. So with this in mind, we then performed a behavioral study to see the consequences of activating or overactivating this ascending pathway. So if we take a mouse that is expressing channel rhodopsin and in the absence of light, monitor the trajectory, you see what you've seen before. There's a relatively direct trajectory to the target with a typical velocity. If we now turn the optogenetic system on and activate and perform the behavioral task, then what you find, I think you can see here, is that there's a severe impairment in the trajectory and reaching. This animal is clearly showing abnormal trajectories, abnormal velocities, and does a very poor job. In fact, when we monitor this, there are no successful reaches in the presence of activation of the PN terminals in the lateral reticular nucleus, and you can see these dramatic changes in trajectory. Now, admittedly, this is a fairly crude approach to this system. We're activating this system in a way that is perhaps non-physiological. Nevertheless, I think it does show that activation of this ascending arm uncoupled from the premotor is producing a severe abnormality in motor control and motor output, in turn raising the question of how is this system operating. We know that activation of the PN terminals activates the LRN neurons, which provide their exclusive output to the cerebellum, and so it begins to suggest a pathway where somehow the cerebellum is involved here in receiving this information and relaying this information. The other important experiment that Eyman and Broer noticed is that when they record from motor neurons under this optoactivation protocol, with some delay, they actually see a series of excitatory inputs to the forelimb level motor neurons from stimulating this PN pathway in the lateral reticular nucleus. And the time delay is not conclusive, but it begins to suggest that one of the outputs of the cerebellar system, perhaps through the reticulospinal, is to provide a descending excitatory input back onto the motor neurons. Now, you could say that we just messed up with the antidromic activation, and what we're really doing is antidromically activating this input. Part of the reason for thinking that that is not the case is that when we sever the LRN input to the cerebellum, we completely lose the motor neuron response here, arguing that this is a cerebellar output pathway. So we're now getting dragged inexorably into the cerebellum, which I think is a bad sign, because no one who ever goes into the cerebellum ever comes out of it. But nevertheless, we're forced into that sort of world. So one way of thinking about what might be going on, and I stress that this is speculative here, is under normal conditions, the activity of these propriospinal neurons under steady state provides an LRN input both to the cerebellar cortex and to deep cerebellar nuclei, and some clever learning strategy, which the cerebellum is good for, ensures that the DCN output is precisely matched by an inhibitory Purkinje-derived signal to compensate for the input from the LRN, and there's a balanced motor output. when we photo-jam this system by activating these terminals, we induce an imbalance. The cerebellum hasn't had time to learn this new world, and so we overactivate these deep cerebellar nuclei, which provide input to the reticulospinal and trigger the motor system. So in some ways, this system for the control of arm movement bears parallels to what some people are thinking in terms of saccadic control of eye movement in terms of the cerebellar involvement. So maybe there are common strategies in motor control independent of whether you're moving the ocular muscles, extraocular muscles, and so we're interested in exploring this possibility by looking in more detail at what the LRN is doing and beginning to delve somewhat tremulously into cerebellar circuitry and output. So just to end, I want to give you one other example of the way in which this reach task might be important and informative in dissecting out, this time not ascending and descending loop pathways, but local circuits that exist within the spinal cord. And this is this idea of their local sensory gates that are modifying sensory input, motor output that controls muscle contraction. And perhaps the classical example of a gate in the spinal cord was identified some 50, 60 years ago by Mike Fortes and Kay Frank, and was studied by Eccles, which is this strange phenomenon of presynaptic inhibitory control of sensory motor transmission. The idea is that the sensory neurons that are forming monosynaptic connections with motor neurons are subject to presynaptic inhibition by a set of GABAergic interneurons. And the classical Eccles study showed that when you can find physiological ways of activating these GABA neurons, you get a suppression of the motor neuron EPSP that is exerted presynaptically. More recently, we've been interested in the specificity. So is this a general population of GABA neurons that are growing out, and some of them just accidentally happen to alight on sensory terminals? Or are there selective subsets of GABA interneurons, some of which are devoted to the task of forming these presynaptic inhibitory synapses? And work that Nick Betley and Julia Kalschmidt did, first of all by visualizing these synapses. So in green, we're looking at a motor neuron surface membrane. This is one sensory terminal shown in blue, and each of these little red boutons here are the boutons of one of these GABA inhibitory synapses. And they showed that this is a highly selective subset of GABA neurons. We have a transcription factor that defines these neurons. And more interestingly, when we take the sensory terminal away, these GABA neurons will not tolerate any substitute synaptic targets. They withdraw their axon and shrivel up. So they're seeking out sensory neurons. We know something about the molecular basis of that recognition. But for what I want to tell you today, the most important aspect is that these GABAergic neurons, unlike all other GABA neurons in the spinal cord, synthesize GABA through the expression of the GAD2 or GAD65 gene, whereas every other inhibitory interneuron expresses GAD1 or GAD67. So GAD2, together with this transcription factor, gave us in principle a molecular entry point to this precise subset of inhibitory neurons that form presynaptic contact. So together with Josh Huang at Cold Spring Harbor, we got hold of a GAD2 iris Cree mouse, used adenoviral-associated vectors to express this same channel rhodopsin fluorescent protein fusion. And if we look at a single sensory terminal, we can see that this fluorescent protein nicely studs the presynaptic boutons sitting on this one terminal, all of which co-express the GAD2 gene. So we can reliably, with viral infection, express about 80% of these GABA pre-boutons on sensory nerve. So we have a way in principle now of activating the GABA presynaptic neurons selectively. So when we take a hemispinal cord preparation, and this is work by Andrew Fink, a graduate student in the lab, shine light on these neurons and record from them, then light very effectively entrains these neurons at frequencies between 1 and 50 hertz in a linear manner. So we now have genetic access to this selective subset of GABA presynaptic inhibitory neurons. And when we record from identified motor neurons while stimulating sensory input and then activating these inhibitory neurons optogenetically, we can see just what Eccles saw, a reduction in the EPSC elicited by sensory stimulation. So this proves that these transcriptionally molecularly defined interneurons are the interneurons responsible for presynaptic inhibition. So in the 60 years that have followed the identification of these neurons, we still don't have any real idea about the behavioral relevance of presynaptic inhibition, because pharmacology doesn't distinguish these from all the other GABA interneurons. So in principle, presynaptic inhibition is kind of good because it selectively blocks one of the inputs to a postsynaptic neuron, whereas postsynaptic inhibition shuts off the neuron to all of the many inputs. But what is the behavioral consequence of that? So Andrew set out to try and eliminate just these presynaptic inhibitory boutons and examine the consequence for motor behavior using this skilled reaching task. And so he could do this, again, with this diphtheria toxin receptor trick, using Cree driven by GAD65 or GAD2, express diphtheria toxin receptor in these neurons, let the mice recover, and then at some point apply diphtheria toxin. So pre-diphtheria toxin application under these conditions, you have nice GABA pre-boutons in pink studying these blue sensory terminals. After you deliver diphtheria toxin, you completely denude the sensory terminals, but all the postsynaptic inhibitory boutons on motor neurons and other neurons are still intact. So this gave Andrew the ability to ask, is there a behavioral consequence of the elimination of presynaptic inhibition, or is this system simply redundant with the very prominent postsynaptic inhibition? So the first thing that he found in these reach tasks is that soon after application of diphtheria toxin, the success of these reaches dropped dramatically to close to zero. So something is going wrong. And if one does the kinematics here, again, this is the prior to the administration of diphtheria toxin, you can see a relatively normal trajectory from origin to target. What about after eliminating the GABA terminals? So this is a mouse in which just that set of GABA inhibitory interneurons and their presynaptic terminals have been eliminated in a relatively acute manner. What you can see, I think, is there are profound reaching defects. The animal is sort of moving backwards and forwards, explains why an attempt fails here. There are essentially no successes under these conditions. And when you look, what you see is, again, a very dramatic behavioral consequence during these reach tasks here. And in fact, if you monitor velocity, you see this sort of oscillatory behavior, which I think you saw in the video. Every forward movement is compensated by a backward movement there. And these have, if you do the transformation, periodicity of 20, 40, and 60 hertz, whatever that means. So getting rid of GABA presynaptic boutons produces this defect in behavior. And Andrew, together with my theoretician colleague Larry Abbott at Columbia, have been thinking about this in the context of what the role of presynaptic inhibition might be in terms of the control of motor behavior at a given limb joint. And this analysis that I'll show you sort of echoes some earlier work that Hans Hultborn had been thinking about in terms of studies that he'd done in cat. And so the idea here is to build a model in which we consider a single joint and the angle of extension and the torques driving extension, which increases this angle, or flexion, which decreases it, and then also to introduce a component of sensory feedback and its presynaptic inhibitory weight. And so the basic idea here is that if we're going to extend a given joint here, activate the motor neurons, activate the muscle, there will be a compensatory, presumably, sensory activation of the antagonist flexors. And that will then feed back in the form of a monosynaptic connection to the antagonist motor neurons and produce an opposite behavioral response. So in this view, one role of the GABA pre-inhibitory neurons is to shut off the antagonist input to motor neurons under conditions in which you want to achieve a smooth pursuit, if you like, of this joint extension. So when you modulate, you base this, and then you simulate joint extension under the conditions that GABA pre-neurons are active, you see a nice smooth extension. What happens then, as we did experimentally, if you take this model and try and eliminate these GABA pre-neurons, under those conditions, there's nothing stopping this sensory feedback from activating the antagonist flexor at a given joint. And if you model this, then what you find is that you have a system in which the joint begins to extend, and then it reverberates in this oscillatory manner because there's this alternation of activation of flexor and extensor and flexor and extensor. And if you plot this, the simulated trace in the presence of presynaptic inhibition and in the absence, if we look at the actual data here in terms of a distance to pellet, but you could measure many other things, you have an oscillatory behavior that, in principle, could suggest that one of the functions of these presynaptic inhibitory neurons is to prevent the consequent reverberation at flexor and extensor systems at a given joint. So for this study, then, it begins to point how understanding the genetic provenance of one set of inhibitory neurons can reveal something that so far was not appreciated previously. And the real question now is that if, by extension, the extensor activation is blocking the flexor sensory afferents, there has to be a high degree of specificity in the link between the extensor input and the inputs to these GABA-pre inhibitory neurons. And Pablo Ruderman's work over many years has argued there's a highly sophisticated organization of presynaptic inhibition in the spinal cord. So we're now interested in chasing that down to see whether this logic can be moved one neuron upstream or two neurons upstream of these last order presynaptic inhibitory neurons. And we can do this these days because we have genetic access to these neurons, can hop retrogradely using rabies virus, and try and trace the neurons that are providing this input. So here the idea is to look at these local sensory gates that are having rather a profound effect on the fidelity of movement and see where that gets us. So just to finish, I've told you a lot about the intricate details of circuitry in the spinal cord and the way that one can begin to use genetic identity to build up these circuit maps. And a general question that you might ask is this a particular feature of the spinal cord, something strange in the spinal cord, or are there lessons to be learned about the organization and function of circuits in other regions of the central nervous system, for example, in neocortical circuits? And these are early days yet because the information on the identity and organization of neurons that contribute to these circuits is less secure, but I think there are a few things that one can begin to conclude. First of all, in cortex, as in spinal cord, there's a very high diversity of neuronal subtypes. You look at Peter Somogyi's work and Klausberger's work in the hippocampus, and you see many, many different interneuron subtypes. The stereotypic organization of settling position and topographies in cortex are just as evident as in the spinal cord, as we talked about. Work from Jeff Macklis and Sue McConnell and others has indicated that there is a transcriptional determination of neuronal identity, at least in the callosal and descending projection neurons. So these studies begin to suggest that, although it's early, the cortex may use very many of the same principles of organization that have become to be clear in the spinal cord. And then the final thing I want to point out is that the cortex is, famous for its experiential modification. And I don't want to leave you with the idea that the spinal cord is rigidly determined by these molecular probes, molecular programs. I think it's very likely that the details of interneuron connectivity, as you begin to look at finer and finer resolution, will have elements where there is an activity-dependent sculpting of these fine interneuronal organizational connections. But in order to get to that level of resolution, we need to be able to find the interneurons that are potentially substrates for experience and then explore them one class at a time. And we're in the middle of trying to do that. So in general, I think the principles that operate in one region of the central nervous system are going to be very much the same, but perhaps with different weights. So some regions place an emphasis on experience and activity, and others on these molecular programs. And I think no region, and this is a guess, are likely to escape the dual combination of these two sets of programs. So I'm going to stop there and just highlight the people who've done this work. All of the work on position in the spinal cord of interneurons was done by Jay Bykoff with help physiologically from George Mentis and Estelle Drobak. Ayman Azim developed this reaching task in mice and began looking at the internal copies mediated by propriospinal neurons. This has been a collaboration with Bror Alstermark and Johan Jiang in Umeå in Sweden. And we've had lots of input and ideas and polemic from Mark Churchland and John Krakauer, who've been telling us that we're wasting our time, but we're ignoring them. And in fact, invoking them to come and help us a little bit. The work on presynaptic inhibition is that from the graduate student Andrew Fink, together with Josh Huang, and thinking about what presynaptic inhibition is good for is Larry Abbott. And so I'll leave this there, and I'd be happy to take questions. Thanks very much.
Fabulous. We have time for a couple questions. Yes.
I was very interested in measuring the kinematics of the mouse reaching, and I was wondering if there's any clinical implications for about the fact that a lack of presynaptic inhibition induces oscillation.
Yes, we're coming to grips with this rather strange result at the moment. There have been many previous attempts, Ebb Fetz and other people have tried to look at correlates of changes in presynaptic inhibition. I think this is the first time that we have cleanly eliminated presynaptic inhibition and see this behavioral deficit. Whether this has clinical relevance, I think, is an interesting point. There are many apocryphal studies of highly specialized motor programs where motor performance increases dramatically in Danish ballet dancers, for example, where people have studied EMGs, and one of the interpretations of those studies is that something changes about the functional weights of those circuits. And one interpretation of these studies by Nielsen is that the strength of presynaptic inhibition could be modulated in some way. So we don't know yet. These are relatively new and unpublished studies, but they do begin to suggest that there's a particular function of this presynaptic inhibitory control that can't be substituted by the elaborate postsynaptic inhibitory mechanisms.
May I add also that in the case of tremors, it's well known that this is a sinusoidal oscillation. It comes about just from simple circuitry where you're falling back on a proportional circuit control, and that's exactly what we're seeing.
Yes, I mean, there are many things that will induce tremors of descending origin. This is one particular local origin. I think it remains to be seen to what extent many of these clinical abnormalities that look like oscillations really reflect changes in the efficacy and efficiency of presynaptic inhibition. We don't know yet.
Back to what you showed us at the beginning, it seems like the system has two independent molecular ways of creating a matching and redundant map, right? There's a molecular system that creates the map in the motor neurons and another one that makes the map of the innervation from the sensory neurons. Do you have any thoughts on why you have two redundant deterministic molecular systems instead of one molecular system that through experience creates the other one?
Yeah, I mean, these are thoughts and there's no real validity in this, but one way of thinking about it is if you relied on a Sperrian lock and key-like label system, the job of the sensory afferent is to sort through 50 different motor neuron subtypes and choose the 2 or 3 that it really likes and ignore the rest. And we don't know, since we don't know what those recognition systems are, what the limits of specificity in those systems. It may be that this is trying to design in protein recognition a system that is just too complicated. So the way that I think about this positional contribution is that it simplifies the problem for later molecular recognition. If you only have to choose between 2 or 3 or 4 potential target partners, maybe molecular recognition can do a better job of that than sifting through the 1 in 50 needle in a haystack. So that's one aspect of this. The second thing is that I want to emphasize that both of these programs are an inevitable consequence of the molecular identity of the motor neuron. It's just in one case the molecular identity is devoting itself to putting neurons in the right place, and in the other to putting surface labels on neurons. They both may be equivalently taxing in terms of molecular routines and machinery, how to settle in a position or how to recognize, and maybe the nervous system is just in a sense divided and conquered by passing the problem out to 2 different systems rather than I mean, it's not a very compelling answer because we don't know, with one exception, any of the recognition. We don't even know whether recognition systems really operate. It could be that everything is a very elaborate positional grid and that sensory afferents are programmed through some other set of cues to get to that point. So we were surprised by the prominence of position as a determinant of microcircuitry in the spinal cord, and are still working through that phase of bemusement.
Last question here.
What do you think of the implications of this for spinal cord injury, both in terms of re-establishment of connectivity and of driving appropriate motor programs?
You could answer that better than me, Clifford. What I would say is that there are this is in a sense where activity begins to rear its head, because as you know, following high spinal cord transections, particularly in animals, providing active input to spinal circuits has a dramatic impact in motor output in terms of the amplitude and pattern of the EMG from studies of Reggie Edgerton and Susan Harkema. So what is activity doing that is changing the weight of circuits so that you reconstruct effective motor output? So the implications, as I see it, is that if we can really map the organization of the circuits that exist that are controlling motor output in this fine degree of resolution, we have a hope at least of asking the question, under conditions where you're now using sensory input as a surrogate for descending input, what has changed in that circuit? If we knew what has changed, maybe we'd do a better job of enhancing that change under conditions that aren't so reliant on sensory feedback and locomotor treadmill activity. So I think the understanding of the organization of these circuits in spinal cord will have an impact, but it's more in the relationship between the reactivation of circuits after injury than in coaxing descending corticospinal axons to do better, although it may have implications there too.